It is now recognized that anthropogenic habitat disturbance impacts the phenotypes of wild animals. This is particularly a concern when it comes to birds. A lot of animals exhibit plasticity and adapt well to the changing in their environments. The endocrine mechanisms inside the animal work as the connection between their perception of the changing environment and the physiological responses within the animal. This exemplifies phenotypic plasticity, also described as acclimation to urbanization. By investigating this phenotypic plasticity, we can discover what causes plasticity in an organism and why some organisms adapt well during environmental change and persist, while other species do not. This experiment will also offer some insight into evolution with changing environments. An example of a changing environment in behavior due to evolution is that birds in urban environments exhibit a more bold, aggressive behavior. By observing and manipulating this aggression in birds, we can further predict how these birds would respond to urbanization. Urbanization is a change in the environment that has been found to dramatically alter the physiology and behavior of wild animals. Specifically, male songbirds living in urban areas have a tendency to be more bold and show more signs of territorial aggression. This trend has also been noticed in the songs of sparrows. This experiment explored the relationship between urbanization and neuronal activity within a network of brain regions which collectively are called the social behavior network. In this experiment, we captured wild territorial male song sparrows, and by playing conspecific songs for 6 to 11 minutes and then collecting their brains to estimate recent neuronal activation by looking for the immediate early gene FOS and levels of neuropeptide argine vasotocin AVT, which are both involved in social behavior. The lower levels of AVT were expected in the medial bed nucleus of the strial terminals, BSTMs, which is the region of the brain involved in the regulation of sociality. It was expected that the urban males, who were generally more territorially aggressive, would have lower levels of neuropeptide argine vasotocin, as well as a lower FOS expression. Through the experiment, it was determined that compared to rural males, urban males did have a lower FOS expression in the lateral septum. It suggests that lower neuronal activity in the lateral septum could contribute to behavioral adjustments in urbanization of the male song sparrow. It was also determined through the experiment that, contrary to the hypothesis, the urban male song sparrow has high AVT immunoreactivity in the BSTM. Overall, it was determined that there need to be more research done on this topic of neuronal differences across rural and urban habitats because of how important it is to understand the effects of a drastic habitat change that comes with urbanization. These responses to changing environments, and specifically urbanization, are all thought to be a result of complex brain mechanisms. Originally, the brain mechanism we chose to identify that impacted the effects of urbanization in birds was the immediate early gene IEG FOS, which is a rapidly inducible transcription factor that has been identified recently for its neuronal activity in regions of the brain related to the animal's social behavior network. These networks play a role in social behavior and social patterns. This social behavior includes territorial aggression, especially in male birds. We also discovered that the neuropeptide argine phosphatase and the avian homologue of vasopressin, AVP, plays a central role in the socio-sexual behaviors of the male songbirds. AVP and AVT both play a role in the range of homeostatic functions within the bird. AVT expressing cell bodies in the bed nucleus of strial terminals selectively respond to social stimuli and then regulate male territorial aggression. A brief overview of our materials and methods includes the use of what subjects, histology, imaging quantifications, plasma testosterone statistics we used. The subjects that we used were granted to us by the U.S. Fish and Wildlife Services. We used male song sparrows captured from both urban and rural settings. For our histology, we coronally sectioned the brains of the sparrow using a cryostat, incubated the brain matter, and investigated it thoroughly. Our imaging and quantifications included the quantification of immunoreactivity for FOS and AVT carried out by research assistants who sectioned out different parts of the brain, both white and gray matter, and studied sections separately. For statistics, we used R statistical software for the analysis. We also ran a general linear model with habitat as a leading factor.
There was no difference in testosterone levels as a result of type of habitat. Additionally, it was shown that testosterone was not correlated to the number of AVT-like immunoreactivity cells. However, there was evidence of habitat type significantly affecting FOS. Rural birds had more FOS nerve cells than urban birds. There was also evidence of habitat type significantly affecting AVT-like immunoreactivity. It was also shown that urban birds had more AVT-like immunoreactivity cells than rural birds in the BSTM, but not the PBN. The level of testosterone did affect the number of AVT-like immunoreactivity cells. We performed each statistical analysis with R software to determine the relationships between each bird type and physiological differences. Because there could have been confounding variables that determine the number of AVT cells in the brain, we used a linear mixed model to test if the number of cells was a cause of the type of habitat, the region of the brain, or both. Significance was indicated by a p-value of less than 0.05, which is indicated by the bold numbering in these tables. We concluded that there is in fact a difference in neuropeptide levels as well as differing patterns of immediate early genes between songbirds living in urban habitats versus rural habitats. This tells us that FOS expression relates to amount of social interaction since it led to differences between rural and urban sparrows. Additionally, since higher levels of AVT immunoreactivity were found in urbanized birds, this feature also correlates with social behavior differences. The most important finding in this study was that urban sparrows had less FOS neurons than rural sparrows, which combined with other studies of different animals that support this claim, increased our argument that this part of the brain changes as a result of habitat. In general, it is known that higher levels of AVT in the brain is correlated with decreased aggression in sparrows. However, our findings showed that higher levels of AVT were found in urban birds over rural birds, and urbanization tends to lead to higher aggression. Therefore, our results are opposing to our prior beliefs in past studies. Overall, our experiment showed that differing levels of AVT and FOS neurons is associated with social behavior, which was shown in the different ways that playback was perceived between rural and urban male song sparrows. Some error in this experiment could have occurred in the amount of time the sparrow songs were played back. Since the playback only lasted 10 minutes, while most other related studies played for 30 minutes, this could have affected results. Additionally, this playback could also have inadvertently attracted more aggressive sparrows that were able to be captured faster, and this could have also affected results by reducing variation of behavior. The implications of this research are very significant with the rise of urbanization around the world. It is important to understand how habitat differences affect the neurophysiology of birds so we can better anticipate how increased urbanization will influence the behavior of not just birds but all wildlife. We can use the evidence found in the study to force change in society and policy to protect these birds whose physiology is being transformed as a result of habitat change. Overall, this study does well at identifying and testing the appropriate regions of the brain responsible for social behavior, as well as outlining the consequences that changing habitats can have on sparrows. However, this study could be improved by more repetition of trials, as well as comparing other neuropeptides and genes in other parts of the brain associated with behavior to see how habitat affected those. Additionally, since their AVT results did not match up with previous knowledge in past studies, more research needs to be done with the AVT factor to determine its true relationship with aggressive behavior.